Micronutrient Digestion by Ethan Simpson, Dominic Alejandre, Kevin Clerkley, Samantha Shell, Jessica Malcolm, and Gabriela Iniguez. We will cover the complete path of digestion and absorption of macronutrients along each major site of the GI tract. Here we have a diagram that traces the path of digestion through the mouth, stomach, small intestine, and large intestine. We will break down each macronutrient of carbohydrates, lipids, proteins, one by one, and follow its own path through the digestion process. First, we have carbohydrates. Digestion begins in the mouth during mastication through the action of salivary amylase from the parotid and submandibular salivary glands. This salivary amylase begins to break down complex carbohydrates into simple sugars of disaccharides and trisaccharides. Salivary amylase usually works from about 1 to 2 hours before a change in pH renders enzyme activity. The digestion process continues along the esophagus, which is used as a transporting alimentary canal from the mouth to the stomach. No absorption occurs along this tract. Once passed through the esophagus, broken down particles reach the stomach where more mechanical digestion occurs. Here, stomach contractions churn and mix grand secretions with partially digested food called chyme. Mechanical processes of stomach acid also prevent further chemical digestion of disaccharides and trisaccharides. The small intestine becomes the next site where digestion occurs, as chyme containing disaccharides and trisaccharides are pushed into the duodenum. In the duodenum, which is the beginning portion of the small intestine, pancreatic alpha amylase breaks down remaining complex carbohydrates. Any disaccharides and trisaccharides at this stage are not broken down any further until they reach the intestinal mucosa. Once they come into contact with intestinal mucosa, brush border enzymes of the intestinal microvilli, which line the small intestines, break down disaccharides and trisaccharides into monosaccharides prior to absorption. Enzymes maltase, sucrase, and lactase are all released from the brush border of the small intestine and target the simple sugars maltose, sucrose, and lactose. Absorption begins throughout the parts of the small intestine in the duodenum, jejunum, and ileum. Facilitated diffusion occurs moving only one molecule at a time through the plasma membrane. Here, no energy is required during this process, and further diffusion cannot take place in the presence of oppositional concentration gradients. Co-transport functions in the uptake of glucose and also brings sodium ions into the cell. Sodium ions and glucose molecules must bind to carrier proteins before entering a cell. From here, simple sugars are transported into the cell and diffused from the cytoplasm into capillaries of the villus for transport to the liver in the hepatic portal vein. In the large intestine, no absorption or breaking down of carbohydrates occurs, thus digestion and absorption of carbohydrate ends.
I'll now begin to discuss lipid breakdown and how it occurs first in the oral cavity by mechanical work. At first, teeth grind down food. Chemical digestion then takes place as lingual lipase and enzymes in your saliva begins to emulsify the fat. When food reaches the stomach, begins to churn and break down further into tiny fat droplets. Fat droplets then remain until bile salts produced in the liver and stored in the gallbladder mixes with fat droplets in the small intestine. Gastric lipase is an acidic lipase secreted by the gastric chief cells in the stomach. They do not require bile acid for optimal enzymatic activity. When it reaches the small intestine, pancreatic lipase acts on lipids via the pancreas, breaks it down, results in free fatty acids and monoglycerides. They interact with bile salts and form micelles. Chyme enters the duodenum. Hormones signal gallbladders to contract as well. The triglycerides along with absorbed steroids, phospholipids, and fat-soluble vitamins are coated with proteins resulting in chylomacrons. The interstitial cells secrete the chylomacrons into interstitial fluid by exocytosis as well. And most of the chylomacrons diffuse in interstitial lacteals. From lacteals, they proceed into lymphatic vessels and through the thoracic duct. Then finally enter the bloodstream through the left subclavian vein. Only about 5% of bile salts secreted by the liver enter the colon, and only about 1% is lost in the feces. Lipids are especially packed into lipoprotein carriers as well, like I said, into the bloodstream. And the protein shell of lipoproteins contains apolipoproteins, which assist the lipoprotein in its function.
Lastly, we have protein. No digestion or absorption occurs in the oral cavity. Instead, chemical digestion begins in the stomach with the release of hydrochloric acid, which denatures protein, resulting in the unfolding of the protein, allowing the amino acid chain to become more accessible to enzymes. Chief cells in the stomach lining release the proenzyme pepsinogen, which is the inactive precursor to the enzyme pepsin. Pepsinogen comes into contact with hydrochloric acid and converts the enzyme into pepsin, which is responsible for 10 to 20% of protein digestion. Pepsin then breaks down protein into individual amino acids and shorter polypeptides. The small intestine is where most protein digestion takes place. Chyme enters the duodenum and triggers the release of protease from the pancreas. Pancreatic enzymes only digest a small percent of proteins into individual amino acids. The surface of the small intestine release enteropeptidase, which activates the conversion of trypsinogen and trypsin. Along with trypsin, chymotrypsin, and elastase are also released from the small intestine. They all work to break down specific peptide bonds with polypeptides, resulting in the production of dipeptides and tripeptides. Carboxypeptidase is also released by the surface of the small intestine. It chops off the last amino acid of the polypeptide chain, resulting in the production of free amino acids. Intestinal mucosa. The brush border of enzymes of the microvilli lining the small intestines release dipeptidase, an enzyme that breaks short peptide bonds into individual amino acids. These amino acids, along with dipeptides and tripeptides, are then absorbed through the brush border of the small intestine into the cell body, with both facilitated diffusion and co-transport mechanism. Then they are released into the interstitial fluid by facilitated diffusion and co-transport. Amino acids diffuse into interstitial capillaries and are transported through the hepatic portal vein for transportation to the liver. And the liver releases the amino acids into general circulation. As for the large intestine, no digestion or absorption occurs here.